In this video, we will discuss about how we can increase the transduction efficiency of adeno-associated virus. Since in most of the clinical trial, it is important to check any virus such as adeno-associated virus in vitro potency, and the problems in normal cells having with this adeno-associated virus is about its transduction efficiency. So since this adeno-associated virus usually interact with the galactose carbohydrate structures. However, in normal cells, these galactose carbohydrate structures are being decorated by the sialic acids, and because of this, the transduction efficiency of this adeno-associated virus got reduced. So this paper talks about how they have increased the transduction efficiency by removing the sialic acids. So before going directly into the topic, let us understand how these sialic acids are formed. So this, in order to form sialic acids, it needs to have this compound, that is CMP sialic acids, and the CMP sialic acids, when it goes inside the inside the cells using the transporter that is known as SLC35A1, and within this, when it's reached inside the cells, the sialic acids and CMP are then separated. That is, sialic acids is attached to these carbohydrate structures, and this CMP will then again move into the uh, it will then again move into the cellular matrix, where it's interact with this CMP sialic acids formations. So these CMP sialic acids uh, the key key step for the formation of sialic acids within the cells, and the transporter of these CMP sialic acids into and outside the cells is being taken care of by the transporter, that is SLC35A1, and this SL and so now let us come to the main point. So in this paper, what they have done is they have taken the hex cells and knocked down this SLC35A1, so that cells will not be able to produce the sialic acids. in this case so when there are no sialic acids are formed, the adeno-associated virus, the transduction efficiency of adeno-associated virus increased, because now the galactose are exposed and they can be interact with the adeno-associated virus, and this leads to this endosome encapsulations, and thereafter this uncoating of this capsid, that leads to this viral single strand DNA, and that will form this episomal DNA, and the translation of this DNA into the mRNA, and then, as we know that the ribosome will take care of this mRNA to form the final proteins, and these proteins then can be used to check the in vitro potency of this the genes that are involved in uh, present in this adeno-associated virus. So this has provided so the modified hex cells now contains the glycoproteins with the terminal galactose, as these galactose are more prone to interact with this adeno-associated virus. So this modified hex cells are provided as a opportunity or another genetic tools to check the in vitro potency of the adeno-associated virus. And as whenever a new kind of vaccines needs to go for a clinical trial, they need to provide the informations about this in vitro potency. And so before this paper, there were very different approaches are used, and that are not easy to or that are not easy to do. However, in this case, so once the cell lines are produced, they can be used again and again if they have been stored in a proper environment, that is in a liquid nitrogen in the presence of some DMSO. So, I hope you got a basic idea of what they have done in order to make this adeno-associated virus more prone to transduction to these hex cells. And in order to knock down this SLC35A, they use this CRISPR-Cas9, where the guide RNAs are targeting this SLC35A1 to create a permanent cell lines that does not contain this SLC35A1. If you need to understand more about this idea and how they validated their idea, that is in vitro increase in the transduction efficiency of the adeno-associated virus into these modified X cells. You can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.